But all of these things should be taken with a pinch of salt. Some of these things are very informative. Like I said, cancer mutation, especially if there is risk or history of cancer in your family. But it should never be taken as the absolute truth. This is something that needs to be discussed with a doctor, needs to be confirmed with other kind of molecular tests or blood tests or different validation experiments, as well as always remember there is some errors as well. This is not 100%. It is not like 1-0 that if 23andMe tells me I have a BRCA mutation, I definitely have a BRCA mutation, because you can imagine the DNA is so long and complicated. Maybe when they were sequencing it, there was a sequencing error in the machine. Maybe when they were mapping it, there's never a 100% accuracy match. Maybe it was 80% and they told you there's a mutation. So you should follow up with more localized tests to do that. But you do now have the option of sitting at your home and getting your genome sequence and discovering hopefully all fun things about yourself. So all of this thing that I told you, so there is a lot of data that is generated on a daily basis. The raw data file that comes out of one patient's whole genome sequencing, so if I just sequence the entire DNA, is nearly 1 terabyte, which is huge, right? So you can imagine that if you were to do this for 500 patients, if you're doing a large study, or if you're a hospital or you're a lab trying to study this, there is so much data that is being generated. You'll get the raw sequencing file from your sequencer. You will then convert it to a actual file that you can read and see ATCG. Okay, what does it mean? You'll convert it into actual gene level expression. That's another set of data. Imagine you've done RNA sequencing. Sure, you know ATCG. Then you'll say, okay, if from this ATCG I can identify FLT3 gene has 10 copies, some other gene has 20 copies. That's another data set that you have. All of this has led to a lot of data generation, which means we now need people to manage this data. We now need people to make algorithms to interpret this data. We need people that, once algorithms had converted the data into an interpretable form, now ask questions and analyze it, right? And as these new types of sequencing comes out, what I've spoken about today, RNA and whole genome, is only 2 types. There are more than, I want to say, like 50 types of sequencing now available, measuring different parts of your cell. So people have to now come up with new kinds of tools to look at them. The same kind of tools are not going to work, which has led to this entire field. Actually, it's not new, but this field of bioinformatics, which I also am a part of. My work is mostly focused on these two aspects: development of new algorithms and statistics, and analysis and interpretation of data. So for me, if I have a data set which tells me these many 20,000 genes in a patient's genome, in an RNA-seq, those are the expression levels, and I compare it to a normal person, or I compare it to hundreds of other patients with the same disease, what is it telling me about the disease? What is it telling me about this patient? What can we do to make this patient better? Or in my case, I work with ongoing clinical trials. I get data from patients who have been treated with a certain drug versus patients who have not been treated with a certain drug. So then I compare the two and I'm like, okay, what is the drug doing to this patient? Is it really making the patient better? How is it impacting the other pathways and mechanisms in the body? So those are the sort of questions that I answer. So a very specific or focused question that is actually being asked these days is, let's say breast cancer. There are multiple types of breast cancer, which can be distinguished looking at the RNA-seq expression. There are main 3 types of breast cancers, okay? And if I look at the RNA expression, I see some characteristics of that RNA expression, and I can bucket them into 3 groups. I have done this experiment for 5,000 breast cancer patients. And in each group, I have, let's say, 1,000 patients each. So there is some common emerging patterns to each of those groups. Now, tomorrow, a breast cancer patient comes in I don't know which group does this patient belong to, but I want to know, because my treatment therapy depends upon which group this patient falls into. If it's in group 1, I'll give it chemotherapy. If it's in group 2, I'll give it estrogen receptor therapy. If it's in group 3, I'll do something else. So it's very critical for me to know that this new patient that has come in, which group does it fall. Now, I don't want to do RNA sequencing. It's going to take forever. Well, it's not going to take forever. It is going to take a few hours, and it's also going to cost the patient a lot. It is $1,000 is very less compared to 13 billion USD. But not every patient has $1,000 to give, like, oh, yes, please go ahead and do my RNA sequencing. So, what have I done? The machine has identified that of these three groups, what is the most important few genes that I should look at so that I can identify that this patient belongs to group A, group B, and group C. So out of all like 20,000 genes, I've identified 50 genes that are important. And now in the lab, there's a kit, there's a panel that measures just these 50 genes. The patient will go in, give their blood. It will be, the gene expression will be measured only for those 50 genes through a panel. There are techniques to do that, and looking at the expression of those 50 genes, I'm going to run it through a machine learning algorithm, a classifier to be specific, and that will tell me, okay, is this patient group A, group B, group C? And once I know, I'm going to start the treatment therapy for that patient. So that's what we're trying to do. We are trying to predict which patient has what sort of characteristics using the sort of wealth of data we have already. Or that is one way. Or we want to identify new information. The cell is such a complicated machinery. We only look at 20,000 genes and we're done. We're like, okay, sure. But those 20,000 genes are interacting with each other also. And sometimes for a human to interpret that data, it's very difficult. We don't know how they correlate with each other. We don't know if we increase one, what would happen to the other. We don't know if they act as like units, subunits within the cell. And to sort of identify those pathways, to uncover those mechanisms, machine learning also helps, because machine learning, what it does is the machine is trying to identify common features across these thousands and thousands of samples, and look for those same patterns in the other data we give it and come up with new knowledge. So that's the end of my slides. I'm going to stop presenting.